All right, so back to this anti-metabolite class of compounds. But before I show you where we'll discuss how fluorouracil might work, I kind of have to show you like what happens with the folate vitamin or aka vitamin B9, as well as I guess its involvement in nucleoside or nucleotide biosynthesis. So, sort of that story begins with deoxyuridine, which of course is not one of the DNA bases because uracil is not one of the DNA nucleobases, but this is needed for thymidine biosynthesis, and you'll see that in a sec. So how this works is there's of course an enzyme involved, and that enzyme uses vitamin B9 or this methylfolate as a cofactor, and the enzyme is called thymidylate synthase. But the way that works is the enzymes, you might have guessed, the cysteine thiol residue would attack the alpha beta unsaturated system within uridine nucleobase. It's Michael addition process, and then of course, I hope you can kind of picture the flow of electrons there. Like once that adds, right, there would be an enolate next to the carbonyl right there. There would be a negative charge after the Michael addition. And then it's that enolate that can basically do an SN2 attack on this methylene, which will leave behind nitrogen with a negative charge that could get separately protonated. So the end result it would be right this structure right here. The R group is this benzene ring, and the rest of the structure here that I am not showing to save space. And of course, the enzyme could do retromichael. So this is a retromichael cleavage of the thiol, mainly because you could have the alpha proton that leaves, which could push off the thiolate, and then that would get us to the reformation of that alpha beta unsaturated double bond. Oops. And then after that, right, like this, uh, you could have an elimination reaction happening here that would lead to the protonation of this methylene group, which would get you the methyl group, CH3. Notice at this point it's CH2, and then of course that's a thymidine nucleotide, and then the rest of the sort of spent vitamin B9, which can be methylated again for reuse. And so that's how we make thymine or thymidine um, nucleotide. And of course, that is very much needed in DNA synthesis. Now, um, when if you introduce 5-fluorouracil, though, right? Um, I don't know why it's not letting me go back one step, but anyway, if you introduce the 5-fluorouracil, what happens, of course, is that uh, this takes over the price of the uracil or uridine, and then but the structure is similar enough. Fluorine, remember, is a really small halide, and so like if you look at the shape of this molecule, it's really not that much different than uracil, where you have here hydrogen instead of fluorine. Fluorine is really tiny because of the electronegativity. It keeps the atom and the nucleus really tight and small. So the enzyme can mistake it for the regular uracil base, and so therefore the enzyme could attack that same alpha beta unsaturated double bond, and a similar reaction happens. There's the same flow of electrons as last slide by thymidylate synthase catalysis, and you basically get to the same intermediate. The only difference is, remember, in the normal biosynthesis of our thymidine DNA base, right? That atom is hydrogen that could be cleaved off, but fluorine is not going to leave as a F plus. cation. Hydrogen can do that, can leave as H plus. That's a regular proton, acidic proton, right? So that means this is a very stable intermediate that cannot collapse like the last intermediate, and so the thymidine synthase basically gets stuck here. It's forever covalently bound to this uridine analog, and that's where everything stops. Meaning that you will not be able to make any thymidine um, nucleotide, meaning that you're not going to be making any extra DNA, which means that the cell, unless you make more thymidine synthase enzyme, the cell is not going to be able to grow anymore. And of course, that is useful for both, sometimes for like an antiviral application, but usually for anti-cancer application. And that's how this drug, 5-fluorouracil, would work. And at this point, it might be good time for you to take a second to answer this clicker question. So maybe you want to pause the video and then look at the question about um, that sort of asks you about your understanding of the anti-metabolite drugs. So give yourself a minute there. And hopefully you've been able to figure out the answer here and had a chance to pause the video. The first choices they make you want to eat something, and because to slow down the metabolism. Of course, that's not what this means. And the second choice is they inhibit certain enzymes essential for producing energy. I mean, that sounds a little bit plausible. The third choice is they tend to have antibacterial activities. That is maybe theoretically possible if these things act as an antimetabolite for bacteria as well, but that's not necessarily guaranteed. It's here, the last choice is they tend to kill or inhibit the growth of cells, of our cells. And that's really how antimetabolite is supposed to work. It's supposed to sort of kill our own cells, and that's why it's usually used as an anti-cancer drug. And I think I told you about the natural product compounds or the class of anti-cancer drugs that especially oncologists call natural products. And like we said, a lot of other compounds are natural products that are used as drugs. So this is not the only drugs that are natural products, but for some reason this is the class that they call natural products. And those are the compounds that happen to have these really unique, unpredictable shape and molecular scaffold that binds to microtubules and structures like that to stop mitosis or cell division. And microtubules, if you didn't know, those are the like, sort of hair-like structures that sometimes you can see with a light microscope that I'm sure you've seen before, like especially during the mitosis, right? Like those hair-like structures are pulling the chromosomes apart for cell replication. And it's not just for replication, but anytime anything it needs to be transported within the cell, like a protein that was just translated, those things are transported along microtubules. So those are like kind of like a mobile, portable train tracks within the cell. And as you can imagine, if you have to biosynthesize the proteins involved in microtubules structures, that would be pretty costly to the cell. So we actually recycle all the material needed for this. And then these are made from two different types of proteins called tubulin, alpha tubulin and beta tubulin. And then they polymerize together to make these rope-like structures, which is microtubules. And so of course, for cell replication, this is very important because it's how we designate where the chromosomes go, where all these newly biosynthesized proteins go and stuff like that. So you need to be able to recycle old microtubules and form brand new microtubules for cell replication to happen. And the so-called natural product anti-cancer drugs sort of mess with that process. And paclitaxel, or Taxol, as the registered commercial name goes, is microtubule stabilizer. And then you might wonder like why that would sort of mess up the microtubule usage. But remember, these things need to be recycled constantly. So if you stabilize, if this compound attaches to the microtubule so that it cannot get broken down anymore for recycling, broken down as in you take apart each individual bead or the monomer of this polymeric proteins to make a microtubule with. If this parent structure gets too stabilized, you're no longer able to recycle these, and therefore the cell growth also stops. And this might be more intuitive. Compounds like vinblastine or the so-called zinc alkaloids, a microtubule polymerization inhibitor, meaning these tubulins cannot uh, polymerize because vinblastine attaches to alpha and beta tubulins in such a way that it prevents the uh, polymerization. Similarly, other small molecules that bind to enzymes or proteins needed for cell replication tend to be called natural products. Such is the case with camptothecin, which is a topoisomerase inhibitor. And if you may or may not know what a topoisomerase is from your biology classes, but it's the enzyme that unwinds the supercoiled chromosome DNA molecules so that they can be read for transcription process. And that's of course when you make an mRNA copy of the genetic information in the chromosomal DNA. And in order for that to happen, each strand has to be straightened out. So it has to be uncoiled and unpaired from the paired double helical DNA strand, right? So that's the job of topoisomerase. Um, Another pretty common target within anti-cancer drug development is a kinase inhibition. And there's a lot of biological uh, cascades and pathways that need to be turned on, of course, for cell replication. And a lot of those processes are regulated by kinases. A common target of a kinase is any nucleophile that's available within the protein, like tyrosine phenyl hydroxyl group, which is a pretty good nucleophile, as you recall from the protein modification chapter. If you deprotonate that, right, like that's a pretty good nucleophile, the anionic phenoxide. And so that's often a target that can be phosphorylated by kinases. Now, there's compounds that have been invented, developed that binds to kinases, specific kinases specifically, so that they can shut down a certain biological pathway that leads to cell replication, and this is one of those examples. But imatinib, developed in the early 2000s, was kind of groundbreaking in that was purely, I don't know if you can tell just by looking at the structure, it's very nitrogen rich, with very few, in fact, only one oxygen, and mostly flat sp2 aromatic type of compounds. That's pretty typical of a synthetic drug as opposed to natural product derived drugs. And then this is a specific kinase inhibitor, and so it's been very useful. This compound really proved the concept that you can actually synthetically develop these rationally. So they rationally designed a molecule that fits into this specific kinase, which is needed in cell growth. Another class of compounds often used in the last 1 or 2 decades or so, the antibody drug conjugates, or ADCs. I think we've discussed this a little bit in our protein modification unit. But the way these things work is by uh, modulating where the drug molecule gets delivered to by attaching the drug molecule to an antibody that seeks out certain proteins expressed on the surface of cancer cells. And then you use these linker chemistry that is designed to be cleaved once ADC drug is internalized into the cancer cell to release the drug. Okay, so that's an ADC drug, and then it's a type of a prodrug. And one of the payloads used is a natural product called collegiamycin, which is a really amazing type of cytotoxin. But I think we've discussed this a little bit. It does have this sugar part of the molecule, carbohydrate part of the molecule that recognizes a specific DNA sequence that is found in rival bacteria so that this compound doesn't damage its own DNA for the bacteria that biosynthesizes these compounds. Now, the way this works, I think we discussed this a bit, if I remember right, that this compound was isolated from a bacteria found on the side of the Texas highway from a soil bacteria, but it works just like a restriction enzyme in that it cuts the DNA of a particular sequence, damaging the DNA of rival bacteria. And so first it was recognized as a potent antibiotic, but then later on we found out that this could potentially be used for treating tumors. The way this works is there's a polysaccharide and an A glycan, or not the sugar part of the molecule. Polysaccharide, like we said, sort of recognizes a certain DNA sequence. The A glycan, or not the sugar part, that's what the word A glycan means, is sort of considered to be the warhead of this, almost like a molecular weapon. And the way that works is in an environment where there's a lot of nucleophiles, like say DNA bases, this unique functional group trisulfide can be attacked, leaving behind a thiolate anion, sulfur anion, which is of course a very good and strong soft nucleophile. And remember, soft nucleophiles do add to the alpha beta unsaturated double bonds, which happens to be present in the molecule here. So that would do sort of the triggering and loading onto it, which turns this beta carbon from sp2 geometry to sp3 geometry. And that subtle difference in the geometry is just enough to bring these two alkyne atoms just by a hair closer together. So this is on a hair trigger. That slight difference in distance between these two alkynes will trigger a reaction called Bergman cyclization. It's a radical process to form a diradical that then, of course, is reactive enough to damage the nearby DNAs. And so that's how this molecule actually damages DNA and kills the cell. And we could say that the part that does all this action is our pharmacophore. The rest of the molecule, we could say oxophore. Oxophore is sort of the rest of the molecule. Not that it doesn't matter, but the rest of the molecule sometimes only is useful for drug delivery or guidance. Whereas pharmacophore is the part of the molecule that actually does the action in the case of cancer drugs to kill the cell and so forth. And in fact, you could tell that that's the pharmacophore. There's been other natural products found from other organisms and related organisms that have pretty much the exact same kind of diene, meaning these two alkynes that are just far enough apart to not undergo Bergman cyclization, but if they're brought together just by a hair, they can be triggered into affecting Bergman cyclization that leaves a diradical that damages the cells. And those are examples of anti-cancer drugs. And then I'll call it good here. But in our last video, we will explore receptor-based drugs. So mostly like CNS drugs. A lot of them are psychiatric drugs, and how that kind of receptor-based drugs are developed. So I'll see you in that video.